So the last part of the last part of the lecture 11, we were talking about RNA and how you can actually clone RNA, right? Because you can't just use the RNA molecule, which is single-stranded, to clone the gene that you might be interested in. So in order to do that, you need to convert that RNA into DNA, right? Which is double-stranded. So you make a complementary DNA molecule from that mRNA using reverse transcriptase. We discussed ways that you can use this cDNA or the applications of using cDNA that we, in order for us to study gene expression, right? This is not good. Oh my gosh. So let's go with this for now because we don't really have a choice. I'm not sure why my PowerPoint is giving problems. I can work on it all day on its own, but for some reason with Zoom, it's been giving us problems and I have no idea why. But anyway, so let's continue with this slide. So the ways that we can use cDNA is to determine whether or not, to figure out whether or not we can express our gene of interest from eukaryotes in bacteria, right? Bacteria is not able to splice the introns from a pre-mRNA. So if you want to study particular gene function, you want to use the cDNA from eukaryotes and then express that in bacteria. You can also use cDNA to compare between genomic DNA, right? So genomic DNA will be the DNA that have the introns in there, so you can compare cDNA with the intron splice out because it then helps you to map where your introns are located, what is the exon, etc. One of the most common ways that cDNA is used is to look at the levels of gene expression. So looking at mRNA abundance, right? How much mRNA do we have within the cell for a particular gene, right? And this include either a small scale or targeted approach, such as reverse transcription PCR, or a more larger scale looking at all the genes in the genome using a microarray. The way that we go about making the cDNA is first we need to isolate all of the mRNA within that cell, okay? And that mRNA is referred to as total mRNA, right? Because it's all the mRNA that's present. What we're also isolating in the cell is not just messenger RNA, right? We're also going to be isolating ribosomal RNA. We're going to be isolating all these other different types of mRNAs. Or sorry, all these other types of RNAs. So we need to isolate the total messenger RNA. We know that messenger RNA contains a polyadenylation, a string of A's at the end, so it's polyadenylated. So we need to isolate that pool of that pool of RNA, which is messenger RNA. And the way we go about doing that, or the most common way that we go about doing this, is to use a bead. That's what's attached to this bead is oligonucleotides with a string of T's. Okay, so you add this bead containing all of these oligonucleotides, and you mix it with your RNA, and it will bind to the A tails and extract all of those messenger RNA with the polyadenylated tails at the end. Once you capture this messenger RNA with this bead, then you will have this pool, and then now you can get rid of the bead, and you get rid of the bead by either lowering the salt concentration or by applying a really high temperature to break the bonds between these two. And once you have your messenger RNA, you will have this string of T's that you know, okay, if you isolate it, you can use a primer now, right? That will match this string of A's that you've isolated. And you can use this primer to synthesize the cDNA copy of this mRNA using reverse transcriptase. Depending on the application, So this is if we're looking at messenger RNA. Depending on the application, you can also use random hexamers. And what this means is just random primers instead of the oligo-DT, right? The string of T's. Why you would use this? So if you want to isolate other types of small non-coding RNAs that might not have a poly A tail, you can use other types of primers to isolate those mRNAs. Sometimes people like to combine the two because maybe the poly A's might be truncated or something might be wrong. So sometimes people combine two different sets of primers. But in general, you would isolate your RNA using a oligo-DT primer to synthesize that first strand of DNA or cDNA, okay? So once you have this hybrid, then what you need to do is to synthesize the second strand of the cDNA, right? So you have the double-stranded DNA molecule. So depending on the application, you could choose to add a string of G's at the end of the primers. And the string of G's is added by this enzyme terminal transferase, which will add at the end of your cDNA this string of G's. You do this depending on what you want to do later on. Okay, so maybe you want to clone this cDNA into a special vector, or you want to use this, you want to be able to isolate the cDNA, particularly for some reason. So you add this as a marker. So you know that, okay, I can isolate it because I have this string of G's that's added to it. That's not part of the message of the messenger RNA. So this is optional. So what you need to do is then after that, you need to get rid of that RNA. And the way you get rid of the RNA is by adding an RNase, right? Which will chew up the RNA that's there. It's a nuclease. You can also add an alkaline solution, which will also help to separate the bonds, and then you can just chew it out. So this is something again that is either optional depending on the procedure you're using in your lab, right? So some labs prefer to use alkaline, some labs prefer to use RNA-H. It really depends on the system that you're working with and what protocol you're using. So it's kind of either or. Once you have your single-stranded cDNA, you have this string of G's here. You can add now a string of C's so that you can prime it. Or if for some reason you knew what this region is, you can add a primer here and then synthesize along, okay? So the either or could be, is desirable, but the point is you need to make that second strand of cDNA so you have a double-stranded molecule. Once you have your double-stranded cDNA, sorry, depending on what you want to do, you can add primers. So you can add linkers, which are restriction enzyme sites to the end of this molecule. These linkers need to be added to the end of, after you make this double-stranded DNA molecule. The linker is optional if you want to clone this cDNA now into a vector using a restriction ligation method. Then you would need to have the linkers with the restriction enzyme site that corresponds to the vector region that you're going to put it into. So maybe you have your plasmid with a multi-cloning site in the middle, as you recall, and you want to put it into that site, okay? And you know, okay, I have E. coli 1 present in that multi-cloning site, so you need to add this multi-cloning, you need to add this restriction enzyme for E. coli 1 at the end of these primers. You will do a digestion, you have your overhangs, you ligate it into the vector, which has been digested with E. coli 1. And you can just add ligase and it will ligate you. There you've cloned your cDNA representing your gene of interest, right? You can also do this for all the cDNAs, all the expressed genes in the genome. And this is referred to as making a cDNA library, right? So you're cloning with a genomic library, you're cloning all the pieces. You're taking your genomic DNA, fragmenting it into pieces, cloning them into vector, and then you sequence, right? With cDNA, you're making a cDNA library. So you're taking all of the genes that are expressed, right? So this is not just random genomic DNA that's present within the genome. You're including just what's expressed within the genome. Made the cDNA, you clone the cDNA, and you can sequence that. So, and this is something that's quite often used if you want to do a large scale analysis, is to use a cDNA library, because in that cDNA library, you would have all of the genes that that are expressed and all of the genes that will be making a functional protein. The other application that you can use this for, as I mentioned, was to compare your cDNA with genomic DNA to isolate where the introns and exons are present, okay? So this is your cDNA. You don't have any intron in that cDNA sequence. Your genomic sequence will have the intron. And you can actually visually see this, right? So when you sequence your cDNA, you have the sequence of the genomic DNA because most genomes have been sequenced. So you have that information available on different databases. And you can align them, right? So you can do a comparison and align them, and you can see the regions which there's gaps, yeah? And this is how it's usually represented. All of the stars suggest that they're identical. And then you have the regions of the gap where you are missing that intron, yeah? And you can see the 5' splice site and the 3' splice site that's present for those intronic sequences as well, okay? So it really gets to, you can really do this to figure out where your, where an intron is present or not. You can actually also see this you can also use this method to even look at alternative splicing, right? Because when you isolate your messenger RNA, if this is messenger RNA that has been processed, right? It's already been spliced. So if there's multiple alternatively spliced versions, those will also be cloned. And when you compare that to the genome or the genomic DNA, you will see which introns might still be there or not there, right? So it's really also a way for you to look at possibly alternatively spliced events as well. So yes, the cDNA only code for genes, right? Because it's only what's expressed, right? It's what's been transcribed, and only genes are mostly genes are transcribed, okay? So the other way that you can use cDNA, as I mentioned, is to really express your eukaryotic protein in bacteria, right? Because everything we do, so you want to study a gene, you need to clone that gene and make more of it in bacteria before you can do anything, right? We all transform everything that we need to do in terms of studying the function of a gene requires us being able to make more of it in bacteria, okay? And then we can do whatever studies we want in the organism that we're interested in. So because, animal or eukaryotic proteins contain introns, if we were to just clone the genomic version of this gene into a plasmid and then put it into bacteria, bacteria is not going to be able to splice out these introns. It will just make whatever you give it, and you will end up with an incorrect protein, right? Because your protein is gonna either have a stop codon that's present within this intron, might be truncated. So you wanna make sure that you remove the introns first before you express them in bacteria, because the bacteria cannot do it for you, okay? So this is one way, is that you remove these introns, you have the correct cDNA, and then you can clone this cDNA representing your gene, your expressed gene, into bacteria. to make more of it. The other way that we use cDNA is really to measure whether your gene is expressed and how expressed is it relative to something else, right? So all molecular biology labs do this. I guess most research labs that work on gene regulation also use this method. And the method is that you're trying to isolate just your gene from all of the mRNAs that's present within the cell. So when you isolate your messenger RNA from the cell, you make a cDNA, and then you need to isolate that cDNA representing your gene of interest, okay? And you can do this because you know the gene you're interested in, right, from the genome, right? So you know what that gene is. You can design primers to just isolate that cDNA only, okay, and look at the expression of just that gene. This is a quantitative method, meaning you can tell how much, rather than other methods where it's more qualitative, right, where you can visually look on a gel, okay, yes, it's there, no, it's not there. This method, quantitative reverse transcriptase PCR, is really powerful because you really can tell how much RNA you have relative to the other RNAs within the cell. We'll talk, and this is all, again, these are all targeted approaches, right? And you'll see in the last lecture where I really want you guys to understand the difference between just focusing on one gene versus focusing on all the genes that's expressed in the genome, right? Because that's where we are currently in research, right? I mean, we have the ability now to look genome-wide at everything, and it's easier to make connections if you know how all these other genes are behaving under certain conditions or in certain tissues. So this is one method, and you'll see how this method is being modified to use on a much broader scale or genome-wide using sequencing. So, so that's where I want to stop with the expression of the genes. And since we're just discussed this, and it should be very fresh in your mind, can you see this poll? Sorry, I need to figure out how to turn this on. Can you see this poll? Yes?
Yeah.
Okay, okay. Questions are silent, okay. So yeah, so let's just take a minute to go over this. We just discussed this, so it should be on your mind, and you should try to, remember this as well over the next 5 lectures before the final exam. And think of a more comprehensive answer, right? I mean, we've talked about many things that cDNA might be useful for, so. Okay, let's end the poll. Let's move this away just in case I need it. So end the poll. Let's share the results. you can see the results. And again, the majority of you were, were able to get this correct, right? I mean, we just talked about the many different applications of cDNA, and also one clue here for an answer is that it says the applications of cDNA are. So the fact that it's plural suggests that there's multiple options here, okay? So we definitely know region is spelled wrong, my apologies, yeah. So we definitely know that cloning the promoter region upstream of the 5' UTR of a gene, that's not what it's used for, because we just talked about the fact that it's really just used for genes, right? And the coding sequence of a gene. And upstream of the 5' UTR, which is the promoter, or part of that promoter region, is all genomic DNA. So it cannot be A. We've discussed the methods, right? Using qRT-PCR, locating the position of introns, right? Expressing an animal protein in bacteria. So since A is not included, it cannot be all of the above. Instead, it's really just these three options that we really discussed that you need to know. Okay. Okay. So that's just to get you guys thinking about the type of questions that you can see. And the exam, so let me move this out of the way. Now I have a million windows open because this, so you can actually see you guys. Chat, okay. Yeah, so this is really going beyond looking at gene expression, right? So we talked about the fact that you have, we talked about DNA replication. We talked about both transcription in eukaryotes and prokaryotes, how in eukaryotes, how it's being processed, right? The next step is to talk about translation. But before we get to translation, there's other ways that you can regulate the RNAs, okay? And that is a field that I would say it's not fairly new, but there's a lot of unknowns in this field. And this involves the use of small non-coding RNAs, right? So these RNAs do not code for genes, but they're expressed and an mRNA is made from it, okay? And then the second half, towards the end, we will talk about this new technology, which is called CRISPR, for clustered regulated short palindromic repeat. It's a mouthful, but we just refer to it as CRISPR, which kind of is very related to how small non-coding RNAs regulate gene expression. And we'll talk a little bit more about that. So, in the early 2000s, there was a lot of studies, again, looking at gene expression. Genomes were sequenced. We can understand what the genes are and what's happening in the genome. So there were these two scientists who noticed that whenever they were working on C. elegans, which is the worm, and they noticed that whenever they try to express certain types of RNA within the cell, you can have a different outcome, okay? And this kind of led them to hypothesize that there's something about the RNA that they're introducing into the cell that's somehow turning off the endogenous copy of that gene. Okay, so the gene that's present within that organism. So they did an experiment where, in the great thing about C. elegans, or the worm, is that you can just feed it anything and it will just eat it, right? So you don't need to, that's the way you transform it compared to all the other different ways we talked about transforming an organism. C. elegans, you just feed it, right? And it will just take up whatever you give to it. So they did this experiment where they used two single-stranded molecules. One is the sense strand, one is the anti-sense strand. And then they also did a double-stranded RNA, okay? So to look to see, okay, if we were to introduce this into C. elegans, which of these versions is really responsible for altering the gene expression that we see, okay? And so in this experiment, they targeted a gene in C. elegans that's responsible for behavior. And that gene is gene that's encoded by MEX3, but it's a gene that causes C. elegans to twitch, right? So if that gene is mutated, the worm just starts to twitch, right? Again, it's a very visual assay because you can actually look at them twitching on a plate, yeah? So they decided to do this experiment, and when they transform the sense RNA or the antisense RNA, for the most part, the worm was kind of normal, right? So it's wild type. They didn't really see any major defect in its behavior. But when they did a double-stranded RNA of that gene, you see that the worm starts to twitch, right? So this suggests that it was going to be turned off. Similarly, when you look within the embryos of the C. elegans, what you see is that again, expressing double-stranded RNA, you're feeding it into the worm, you look within the cell, what's happening. Here is where it's a control, right? It's an embryo with no anything introduced into it, right? So it's just looking for the expression of the gene. It's kind of a negative control. In B, you have a positive control. So this is a parent that was not injected with the RNA, right? So there's no RNA, okay? In C, you have a parent that was injected with the antisense RNA, right? Which is a single strand antisense RNA. And you see kind of a reduction, but you can still see a lot of it being expressed, right? You can still see this kind of grayish color. So it does, to some extent, the single-stranded RNA does trigger a little bit of reduction, but not completely. And it's only when they injected it with double-stranded RNA that you really get a significant reduction of that gene, okay? Do they twitch if you add both single-strand? No, so you need to have the double-stranded molecule. So that's a great question. So what happened here with this experiment was that in the case of C, they did follow-up studies, which we're not showing here, where they addressed why is it when you had this single-stranded RNA molecule, you get a little bit of a reduction, right? The hypothesis was that you needed a double-stranded molecule in order to completely turn off that endogenous gene. So follow-up studies from this group and others showed that the reason that you had this happening was because there was a contamination, right? So there was a contamination in this pool of antisense RNA, where some of it included double-stranded RNA, okay? But not a lot of it, just a little bit of it, enough to show the reduction or this partial silencing. So this was discovered after, which we know now, okay? So the conclusion from this study and all subsequent studies after that was that this double-stranded RNA molecule was somehow recognizing that endogenous RNA for whatever gene and turning it off, right? So it's destroying the RNA somehow and turning that gene expression off so you can't detect the expression. Similar studies were done in plants. And the idea was that there were groups that were studying flower color, right? I mean, it's a very easy and visual measurement, right? The colors are this color or that color, right? You could easily see it. So there were groups that were studying in plant biology, they're doing research studying flower color. And they were interested in petunia. They wanted to be able to create a more brilliant purple color in the flower. It's more desirable, to consumers. They can sell more plants and more flowers. So they knew the gene that was responsible for this flower color. So what they did is they isolated the RNA from this gene and tried to they isolate the RNA, clone the gene, and try to put it back into the plant to see if they increase the expression. So you're adding another copy of that gene, will you get increased purple color? Okay, and that's what you do, right? You want to see you want to understand your gene function, you want to make more of your gene, you want it to be highly expressed, so you put more of it in the genome, right? So that's what they did, right? And a transgene just refers to you're transferring it into the genome. So it's usually referred to as a transgene or transgenic. And when they put it back into the genome, they got the opposite effect. Instead of having more purple, they had these flowers that had these stripes, these white stripes, right? And all of these white areas are areas where the gene was turned off, okay? So what they found out was that in a subset of these flowers, somehow that what they transferred into that genome was turning off that purple color, that gene that codes purple color in the genome. And this also supported the idea that there is a mechanism in the plant that is triggering the degradation of that endogenous gene, okay, and turning it off, okay? And based on that study and other studies in other organisms, right? We talked about the work we did was shown in worms. Similar studies were done in fruit flies, in Drosophila, and also similar studies were done in fungi, right? So in Neurospora. And what they found was that the mechanism was very similar across all of these organisms. Each researcher, depending on the organism, called them very different things, right? So it was either referred to as co-suppression, post-transcriptional gene silencing, or other types of names. But we know now that it's really the central name that everyone agreed upon was RNAi, right? This is RNA interference. There's something that's interfering with the endogenous RNA. Okay, and that's a term that represents all of these observations that was observed in the many different organisms. So this is what we refer to when we talk about RNAi, right? It's all of these shared mechanisms, and that's what we will talk about for the rest of the course, or for the class. So RNAi is it's a big term, right? Because there's many different mechanisms underlying this RNAi. But the general idea is that you have a double-stranded RNA molecule that gets processed into a single-stranded RNA molecule. This single-stranded RNA molecule is bound by many different proteins and processed by many different proteins. This single-stranded RNA molecule shares similarity to some gene in the genome, right? Which is the target mRNA. And it will find that targeted mRNA and hybridize to it. When it hybridizes, you have a double-stranded RNA molecule, right? When it hybridizes, many different outcomes can happen. Okay, so you can either have a cleavage of the target RNA, so it will be destroyed, right? And that's where, so there's no protein product, you don't see any genes being, you don't see any expression for that RNA. It can also involve translational repression and eventual destruction of the target RNA, and we'll talk about that as well. And it can also force the formation of heterochromatin, right? You guys remember what heterochromatin, the heterochromatin is, or what kind of control it has on gene expression? Tight, yeah, exactly, right? So tight chromatin, right? So heterochromatin is where you have the chromatin is completely compacted, right? So it can also force that region to be heterochromatic and of course silence the gene that way, okay? So these single-stranded molecules are generated from double-stranded RNA. And this is important to remember, okay? So it's generated from double-stranded RNA molecules. And there are three major classes of small non-coding RNAs, okay? They're short interfering RNAs. They are PV interacting RNAs. This got the name from Drosophila. from the gene that encodes PW. And you also have microRNAs or miRNAs. So these are kind of the three major classes of non-coding RNAs that's present across eukaryotes. All three types of short RNAs locate their target through RNA base pairing, right? That's their goal, is to find the target, a base pair with that target, to destroy it in some way or to alter it in some way. What's different between these three different RNAs is the way that they're generated, right? So they're generated from different types of double-stranded molecule. So our understanding of RNAi comes from from the way that an organism deals with an infection. Okay. So RNAi will usually occur when a cell encounters a double-stranded RNA virus. So viruses can be RNA or DNA, right? A lot of them are RNA viruses, right? And like the coronavirus that we're dealing with right now, but you can also have viruses that are DNA, right? Has DNA molecule. And in addition to viruses, you can also have RNAi being triggered by transposons, right? So transposable elements can create double-stranded RNA and then trigger this RNAi pathway. And you can also have a transgene, and that's what we're talking about. If you want to transfer into an organism a gene that you want to study, can trigger this RNAi pathway, right? Because it's an invasion into the cell. And the cell recognizes it as a outsider, or something new is being transferred into its genome. Okay. Once this RNAi pathway is triggered, right? So there's obviously sensors, proteins that sense what's happening in the cell. Once this is triggered, you will have this double-stranded RNA molecule that gets degraded by a protein called dicer. Right, so this is an RNase-like enzyme, RNase, because it chews up RNA, and it processes this double-stranded RNA into smaller fragments, right? So these short interfering RNAs in this case, which are usually 21 to 23 nucleotides. From that short pieces of double-stranded siRNAs that exist, one of those strands get degraded, and one of those strands is used in a combination with a range of other types of proteins. We'll talk about AGO2 in a minute. And these protein with that single-stranded siRNA will then find the target RNA, base pair with that target mRNA, and cleave that target mRNA and degrade it, okay? And this cleavage occurs with another group of proteins called the RNA-induced silencing complex, or the RISC complex, okay?
Professor?
Yes.
So if the virus has DNA as its genetic material, how
some
do we
viruses,
get
some viruses,
yeah.
Yeah, it's in particular in this RNAi pathway, the infections are coming from viruses that have the genome is RNA, so they will only be able to infect that cell with double-stranded RNA.
Okay, so if it has DNA, it cannot infect the cell.
If it has a double-stranded DNA molecule, it's a different process, right? So will be a different way that the cell will deal with that type of invasion. And that's where we'll talk about CRISPR, where that's
different from RNA interference.
Yeah, so, because with CRISPR, right, exactly. With CRISPR, you're having the bacteria is usually infected with viruses that have a DNA genome, right?
So RNA interference only deals with the viruses that have RNA as its genetic.
Exactly. Yeah.
So that we can get this double-stranded RNA.
Right.
Thank you.
Mm-hmm. So, so then you have these processes. The way that RNAi work, and this is a very simplified model. There's a range of proteins that you're not seeing here and you're missing here, but they're not important for understanding the basic concept, right? So you have a double-stranded RNA molecule that's injected into the cell. this protein called dicer recognizes this double-stranded molecule, this double-stranded RNA as an invader, binds to it, cleaves it into small fragments, right? So you have these small molecules that's present there. And this dicer complex recruits additional proteins to form this RISC complex, okay? And together, first they destroy one of these strands, which is not shown here, but just imagine that one of these double strands are destroyed. Together, they deliver one of the strands, which is the guide strand, to the target mRNA and cleave this target mRNA and destroys it, okay? Very simplified process. The delivery of this single-stranded siRNA to the RISC complex, as I mentioned, include many different proteins. So you have a protein, this RISC RNA-induced silencing complex, which include a protein called Argonaute-2. There's multiple Argonauts in eukaryotes. The one that's involved in this siRNA process is Argonaute-2. And the minimal RISC complex that exists is this Argonaute protein by itself and an siRNA, right? Because this is what's going to guide, right, and bind to that target mRNA. In addition to Argonaute-2, you also have two other proteins. You have this dicer protein, sorry, and you also have this another dicer-like protein called R2D2. You can imagine the researchers who worked in these proteins, what might have been their favorite shows, right? So this R2D2 protein is also part of this complex. They bind to that double-stranded siRNA that's initially made and help to remove one of those strands, right, by getting rid of it. And then what's left in the end is this Argonaute-2 protein with this siRNA that then serves as that guide to find the target mRNA, form that hybrid, and destroy it, okay? What you need to know, which is super important, is Argonaute-2 and this siRNA, which is absolutely super important. Yeah. So yeah, Argonaute-2 is the one that cuts the passenger strand in half for that double-stranded siRNA, okay? When it comes to the target mRNA, Argonaute-2 also responsible for cleaving that target mRNA, right? So this is now you have the slicer activity of Argonaute, right? These are different domains within the Argonaute protein. So they're not different proteins. This is all Argonaute-2. These are different domains within Argonaute-2, okay? Within Argonaute-2, the PV domain is the domain that recognizes and cuts the target mRNA, okay? And destroys the RNA. And the dicer is initially, so the dicer protein is really what cuts that first double strand, that invading double-stranded RNA, right? So then once you have this complex, right, with this double-stranded RNA, small double-stranded RNA, okay, so you got it from some, yeah. So then you have to go through this step to get rid of one of the strands, right? And this involved Argonaute-2 and the two other dicer-like proteins. And then that complex then finds that target, domains within that protein will then cleave it, okay? So trying to, we're not going to talk about, I mean, the mid and N-terminal domain, they're all responsible for positioning. What's really important is this PV domain that really binds to the target mRNA and actually cuts it at this position. Yeah. And then you with Argonaute-2 is able to cut that target RNA, right? Using the siRNA as a guide. And they actually did experiments like everything else we've discussed in this course to really confirm that, right? So they wanted to look at the activity of the Argonaut-2 protein from humans. So they did an experiment where they just did really, it's an in vitro assay, right? Where you have the Argonaut-2 siRNA, you isolate RNA that targets the particular gene you're interested in, mix it together in a test tube and see whether or not it's able to do its slicing activity. And then you run a gel to look for the fragments, okay? And this is all RNA, it's labeled. You're looking at a jelly electrophoresis as usual. And what they did is this particular enzyme also needs a metal ion in order to work, right? It's required for the activity. You remember Taq polymerase, sometimes you use magnesium with Taq polymerase. This is also an enzyme which also requires a metal ion for proper functioning, and that's magnesium. So these experiments, the first 3 lanes are showing you an siRNA that recognizes this target mRNA in this region. So when you cleave, you have 2 different fragment lengths. There's another siRNA, siRNA2, which cleaves in a different region of this target mRNA. And when that cleaves, you will also get 2 very different products, okay? So here you're showing siRNA 1 or 2 or no siRNA, this is your control. And then on the right-hand side, you're looking exactly at the same samples that's here, except that there's no magnesium chloride added, right? So this not only tells you that you need magnesium, right? Because nothing is happening here, but also that this Argonaut-2 is able to perform that slicing activity. So with 2, you have 2 fragment bands, right? You have this product that's here, and you also have a smaller product, which we knew what those sizes were. And then you have a different fragment size if it's these 2, okay? So exactly, so it's really very much like restriction enzymes where you're cleaving the mRNA molecule into a smaller fragment and destroying it, right? So that confirmed that Argonaut-2 has the slicer activity. So the question now becomes where, if you're making these siRNAs from a double-stranded RNA molecule that just invaded the cell, how do you continue to silence the target mRNA, right? Because the gene is there, it will get expressed, it will be transcribed again, right? And you will, it will, if it's a gene that needs to be turned on or is always turned on, it's going to be transcribed, right? How do you keep silencing or degrading that messenger RNA? So the way that this is done from that initial double-stranded RNA that you have is that the cell is able to keep amplifying these siRNA molecules. And these molecules are, more and more of these siRNAs are made during the process of this RNAi pathway. Okay, so some of them some of the siRNA will hybridize to the target and degrade it, while others will hybridize to the target and synthesize in antisense RNA that then becomes a double-stranded RNA. This process is generated by this RNA-directed RNA polymerase, which is different from our other RNA polymerase, right? So our other RNA polymerase, RNA-Pol2, is a DNA-directed RNA polymerase, right? Because it's binding to DNA and making RNA. Here, this is RNA directed because it's binding to RNA and making RNA, okay? The new double-stranded RNA that's then made from this process is then digested by dicer, and you have a new piece, and this cycle continues. And that's really what's shown here in this amplification model, right? Where you have your double-stranded RNA, which just triggers this whole process. Dicer dices it up into little double-stranded molecules. You go through the whole process we talked about, getting rid of one of the strands. And with these double-stranded RNAs, you then are able to, for some of them, will bind to the target mRNA, cleave the target mRNA if possible, right? That's the step we just talked about. But then some of these single strands will bind to that target mRNA and synthesize a strand, okay? So it will use this as a primer, this RNA-dependent RNA polymerase. It uses this as a primer and just synthesizes it, yeah? It's able to do that using the antisense, of course, because it's 5' to 3'. It works the same way in polymerase and synthesize a double-stranded molecule. When you have this double-stranded RNA that actually came from this initial process, that your new double-stranded RNA molecule dices it up into little pieces. It goes back into the pool of the cycle, and this is the cycle that gets repeated, amplifying these siRNAs. That some of them will now target the target mRNA and destroy it. The others are going to remain to repeat this process over and over again. Okay, so this is how the cell makes sure that if it's targeting a gene for silencing, it makes sure that that gene remains silenced. And we know that all of these proteins are important because in mutants where you delete or alter the protein in any which way, you affect this RNA pathway using siRNAs. So in addition, we mentioned that RNAi can help to trigger heterochromatic regions, right? To amplify those regions. And the way, I said, see the next slide. So in the way that this is done is by making that chromatin area around a particular gene of interest more compacted, and therefore you cannot really access the region and help to transcribe or help to make more of that mRNA, okay? Because the mRNA is already made. So imagine that you have this double-stranded RNA molecule, which is cleaved by dicer, right? You have your double-stranded siRNAs. This is the pathway we just talked about, right? You have your race complex comes in, finds your target mRNA, destroys it, right? RNA-induced gene silencing. Alternatively, what can happen is that you can have some of these double-stranded siRNAs that gets processed. Another complex called RITS instead of RISC, right? Which is RNA-induced transcriptional silencing here. No, instead of RISC, sorry. So this RITS now then targets this mRNA. And when it targets this mRNA, it's able to recruit histone methylate. It's able to modify and recruit histone modifying enzymes that will recruit chromatin remodeling complexes that are involved in gene silencing, right? So the DNA methylation will occur, histone methylation will occur, and they will recruit other complexes that are involved in gene silencing, okay? So such as methylases, right? So it will recruit the DNA methylases and methylate this region.
And that would be, yeah, sorry, that would be the DNA of the host or the
yeah,
host.
Yeah. So it's kind of helps to recruit all of these chromatin remodeling or epigenetic regulatory methods and just silence that region. Okay, this happens a lot for transposons, and this is kind of the mechanism that is used for silencing transposons that generates a double-stranded RNA. There's another method to help to silence that's referred to, that uses, instead of siRNAs, it uses these pi RNAs or PBRNAs or PBRNAs. And these ones are very effective at blocking transposons, particularly in the germ cells, okay? You don't want transposons moving around in your germ cells, right? Because if these are the cells that gets inherited, right? They give rise to gametes, they get inherited to the next generation. if within these cells there's a mutation, these mutations get transferred into the next generation, which becomes problematic. So particularly in germ cells, you have silencing of transposons using this PV interacting, this piRNA mechanism, okay? And these piRNAs in general, they are usually made from pi proteins that's present within the cell that share similarity to a particular transposon, right? Some aspects of it share similarity to a particular transposon. They're found in many organisms, and they are really precise in making sure that that transposon gets silenced in the germ cells, okay? The size is very similar to siRNAs, right? 24 to 30 nucleotides long. And often you will find complementarity to some part of the transposon. It's similar to what you have with the siRNA with your dicer proteins and that RIS complex. These proteins are bound to the target mRNA with the help of PV proteins and another type of protein called abergine. It will similarly base pair to the target mRNA and cleave the target mRNA, okay? This includes a series of steps that in addition to cleaving the mRNA, you also need to process the 3' end of that target mRNA. This is important for also kind of amplifying this process. Okay, similar to how you have the siRNA being amplified through that triggers that mechanism using a RNA-dependent RNA polymerase. This 3' processing is important for amplification of this step. So in very simple form, right, you could imagine that you have a pi RNA. Pi RNA have a very special feature, right? It has an RNA, it has a U at the 5' end. This is how it's made, right? And you'll see here in a minute how that's how it came from. So let's just start here. So you have this siRNA, not siRNA, sorry, pi RNA with the U at the 5' end. This will share similarity to a transposon mRNA. That is the target mRNA, that transposon, okay? It binds to the target mRNA with the help of these PV abergine proteins, which within siRNA would be like your RIS complex and your organelle 2. So what it does is that it binds to the target. When it binds to this target, this protein specifically cleaves 10 base pairs downstream of this target A, right? In this target A. It cleaves at a particular location. So it's very precise. It will cleave, the pi abergine will cleave here. And in addition, on the other half of this target mRNA, where this A is bound to this U from your pi RNA, this 3' end also gets processed, okay? So you're left with this fragment when this cleavage occurs, okay? This fragment of this transposon is then what is used in this step to amplify this process again, okay? So it cleaves, it gets destroyed, but part of it goes back into this complex bound by another Argonaute protein, Argonaute 3, right? This protein then recognizes a group of, again, it came from there, so there's other sequences that are shared that's present within the genome. It will bind to other pi RNAs that's present within the genome that shares this similarity, cuts it specifically adjacent to this U, right? And cleave this molecule. And this is the molecule that's used to start that process all over again, right? So this is just inverted, right? So 5' to 3', you have at the 5' end this U. So this is the cycle that you have with these pi RNAs, where you use them to silence the transposon and then use a piece of that was cleaved or degraded to re-amplify this process and continue to cycle this method. So it's called ping pong amplification because you're using one to hit you're kind of back and forth. So you're using one to silence the other, and then it's coming back, and that's what you're using. So it's kind of, I guess if you think of a ping pong, right, is where you're kind of you're hitting something and it's coming back to you, right? So you're cleaving this target mRNA, and that's what's being used again, right, to go back. So, and then ping pong is kind of a weird name because there's particular transposons that are actually called ping and pong in plants. So in this case, this is just based on kind of the mechanism as they see it. So that's with the pi RNAs and how they really target transposons. The siRNAs degrades the mRNA form and also kind of forces heterochromization or heterochromatic regions. So then with microRNAs, which is another class of small RNAs that participates in gene silencing as well. These are about 22 nucleotides in length, and they're naturally produced in plant and animal cells. They are produced in the cell from larger RNA precursor that happens to fold back on itself and make a double strand and make a kind of a hairpin structure. Okay, because we know that single-stranded RNA is very unstable. They're really prone to making all of these secondary structures and folding back on itself. And these microRNAs actually comes from some of these larger stem loop type of RNAs. In animals, these microRNA will base pair with a 3' UTR of some mRNAs and silence the gene by blocking translation. Okay, so the ribosome is not able to continue. In plants, microRNAs base pair with mRNAs and they actually direct the cleavage of mRNAs. The process by how these work, which we're not going to, again, it's very simplified, I'm not going to go into too many details, is imagine that you have your large precursor the large precursor RNA, right, which is kind of the stem loop, double-stranded, you kind of have base pairing here. Also gets processed by dicer, yeah, in this case. Then you have your small single-stranded microRNA after all of the processing steps. Depending on where they bind, you will have different outcome, and also depending on the organism you're looking at, right? So it will base pair in this 3' UTR region in animal cells, right? When it base pairs here, it blocks translation, right, of this mRNA. In the case of plants and also in some animals, the mRNA will actually base pair within the coding sequence or the express region of the gene. And when it does that, it will form that cleavage that you see and cleave the RNA. So either of these two outcomes can happen. Particularly in animals, both of these processes are possible. In plants, this is the only process that's possible, right? Where the mRNA gets cleaved and destroyed. So these small RNA, all of the small RNAs we've spoken about so far is, again, it's really an active field. There's still a lot we don't know about them. There's a lot of research going into studying small RNAs because they really can regulate gene expression. And the techniques are becoming more and more easier for us to be able to study small RNAs compared to in the past where it was really difficult to look at small RNAs. It requires using actually northern blots is a very popular way to study small RNAs. But with that, you have to use a lot of radioactivity, right? So, and that's one of the disadvantages of northern blots. You have to use radioactivity and really lots of radioactivity. So no one likes to do that if they don't have to. We have right here in our college, Shumei Chen, who is a professor. She's in plant biology, but she studies small RNAs, and she's really an expert in the field in small RNAs. She's in the National Academy of Sciences. very famous. And so a lot of her work, a lot of the breakthroughs that we know on small RNAs came from a lot of work that that her lab has done. So that's small RNAs. So, you know, if you think of that technique, it is we knew that this existed in eukaryotes. We have also leveraged this mechanism to study gene function, which we didn't talk about, right? How can we use this mechanism of small RNA silencing to study gene function? So we're able to do that, okay? More recently, always within the last 10 years, I would say, we discovered a different pathway that bacteria has been using to destroy invading viruses. So this technique or this mechanism is called CRISPR, right? And it is a way that bacteria and archaea bacteria, very early bacteria, use to prevent infection by viruses. It's considered the immune response in bacteria, okay? The reason it's considered immune response is because once that bacteria has been infected by a virus, it will always remember it, right? So it's kind of a vaccination. When we get a vaccine, right, you get infected, you have the vaccine. If you get infected with that virus, your vaccination that you have will recognize, oh, this looks familiar, let's get rid of it, right? The same thing with bacteria, and that is what this entire idea came from CRISPR. It's a very fascinating field that's really moved very quickly. So the idea being in bacteria, if bacteria is infected by a DNA virus, right, which most times it is a DNA virus, what it does is that it immediately cleaves this viral DNA. It then takes this viral DNA and inserts it in a region in the genome to keep it as a memory, okay? It also puts this piece of this virus in a particular region that is referred to as the CRISPR locus, right? And this particular region, because again, it's clustered, right? Clustered, regularly interspaced, short palindromic repeats, right? Because they have these repeat sequences that are adjacent to these the piece of RNA. So all of these green and brown and blue are past infections that has been placed here. So every time a new virus comes in, it goes to this 5' region. So you can say, okay, this is the newest virus and this is the oldest virus. Bacteria, right? Pretty brilliant in my mind, right? So it has this region to remember. When this region gets transcribed, it makes an RNA, which is called a pre-CRISPR RNA, right? This pre-CRISPR RNA gets processed, right? So each of these individual pieces gets packaged with the help of a protein called Cas protein, right? This protein, there's many of them, but this Cas protein binds to each of these. And then you have these CRISPR RNA molecules that's floating around in the cell. If that same virus comes in, if the bacteria gets infected with that same virus, you have this similar sequence with this protein already present. It will find it and destroy it, okay? So it doesn't need to put it back into this locus because it already have it. It's absolutely brilliant. I mean, it's really a clever assay. Of course, we as scientists want to find a way to use this to our advantage, right? So in the course of studying this mechanism, it was noticed that the similarity is very similar to the RNAi mechanism that is used in eukaryotes, right? You have a foreign invasion, right? CRISPR, it's foreign DNA. RNAi is foreign RNA, right? The difference here is that with a CRISPR, the RNA gets integrated into the genome. The DNA gets integrated into the genome, my apologies. With RNAi, it's not integrated into the genome, right? It comes in, it gets attacked by these dicer. Here in bacteria, you have this Cas protein that helps to cleave this region and also bind to it, right? Forming this RNA protein complex, right? Similar here, dicer forms this complex, right? Find the target, destroys the target. So very similar idea. What's different is the source, right? It gets integrated, the proteins that cleave them, and then the target where it targets the RNA, okay? So this is a very shared process that was observed, and it was like really interesting because it's like, oh, this is very similar to what we see in eukaryotes with RNAi, but because this is in bacteria, maybe we can use this system in bacteria to study gene regulation in eukaryotes. That went very quickly in terms of how can we adapt this system to use in eukaryotes. And what we found was that you can literally use this CRISPR system to edit any genome that has been studied, regardless of where you're looking, eukaryotes, prokaryotes. You can use this system to edit the genome. It requires several features, okay? And again, to date, I mean, I think if any research lab that's studying gene regulation has either used or plan to use or is using this technique to alter the genome in some way. And it's a way that we are actually able to make transgenic organisms very easily, particularly plants, right? We've had issues in the past with regulation from many different sources, right? In creating genetically modified organism. The way that CRISPR works, it's really not considered a genetically modified organism, right? And I'll explain what I mean by that in a minute. So in order for you to be able to use this CRISPR technology for anything, you need to be able to isolate some features, right? Remember, you're not using this in bacteria, so you need to have features that you know will work in any other system. The basic strategy include this Cas9 protein. I told you there's many different Cas proteins. Cas9 is the main one that we use. This protein is expressed in cells along with the guide RNA, right? Because remember, you have that protein, you have this guide attached to it, and that's what's going to find, you know, the DNA that you're trying to find to cleave it, right? So if you have this Cas9 and guide RNA, which you can say, okay, I want to target this gene in the human genome that I know codes for, let's say, eye color. You have your Cas9 protein, right, where you can order it. You have the guide RNA, which is a piece of RNA representing that gene that you're interested in. You assemble this, right, in a test tube in your lab, right, doing PCR, etc., and then you put it into the genome. Okay, this Cas9 and RNA protein will then find that target where that RNA is similar to and cleave that target. Okay, in order for that to happen, and that's what's also pretty beautiful about this system, it's very, very specific, right? In order for this to happen, it needs to not only find the target DNA, right, this RNA, which is similar to the DNA in the region, it needs to occur upstream of a particular site called a photospacer adjacent motif. It's 3 nucleotides, that is N, so this N can be any of the nucleotides, GG. Okay, so these 3 nucleotides needs to be present, and then the region that shows your guide RNA. This is the only way that this Cas9 protein is able to cleave that region. It needs to recognize this PAM site. So again, this is all through discovery because this PAM site is not present in bacteria. We know it's present only in the invading DNA, and that's how it was discovered, right? And the reason that we also know this is because if this PAM site was present in bacteria, then bacteria would cleave its own DNA, right? So this PAM site is important for targeting the guide RNA and also for the function of the Cas9 protein. And it's the way that bacteria has found to distinguish its own DNA from invading DNA viruses so it doesn't cleave it. Absolutely clever. It's a beautiful system that we are now able to use to manipulate whatever we want in any genome that we want. Yeah, I'll stop here simply because this is a very exciting topic and I really want to go through the remaining slides in a little bit more detail. So I will stop here and take any questions. I'll be around for a little bit and we'll just continue on this CRISPR later. And we'll do some more polls, which I didn't get to later on in the next class. Any questions? So we can use this technology for, you'll see the many different applications on Tuesday when we talk about it, that's what you can use this for, yeah? Many different applications, yeah. Thank you.